Good morning, all. Welcome, or afternoon, depending on where you're located. Welcome to the ImmPort monthly webinar series. This particular presentation is entitled Fair for Fair Sharing of Immunological Data. And obviously the topic will be focused on what ImmPort does or can do to help you with your fair sharing goals. A few housekeeping things at the start of this call. Those of you who are participating or attending this call, please feel free to use the Q&A to enter all your questions. We do have Zulano, who will be monitoring on behalf of the ImmPort team, and she'll let us know when there's questions from the participants. Okay, let's get moving forward. And on an aside note, we are recording this call for future sharing, so just please be aware. Okay, so the agenda for today is posted right now. We'll just do plan for just a couple minutes of introductions, and then we'll jump right into the presentation for today, followed by approximately 15 minutes at the end for Q&A. But again, please feel free to put in your questions into the Q&A fields. I would like to thank the ImmPort team right at the outset of this presentation, without which ImmPort would not be what it is today. Our PI, Dr. Tolbute, out of the University of California, San Francisco, is, he and Sanchita are the scientific heart of the ImmPort program. So thank you very much. And Ruben Sarwal has been a great addition from the UCSF team, advancing a lot of the scientific efforts on behalf of the ImmPort team. Stephen Kleinstein from Immune System Sciences is key to a lot of the science foundation behind ImmPort, and we very much appreciate the efforts on his behalf. Anu Gubaraj, Dr. Quan Shen, and Dr. Dawei Lin have all contributed. They are program officers for ImmPort and have been with the ImmPort program for many years, and we appreciate all their support. Addition to the folks supporting the ImmPort program are the Periton team. Morgan Crafts is our program manager, and the rest of the team is listed there below. I'm not going to introduce each and every person, but I just would like to extend heartfelt thank yous to each person on the list. ICF is also contracted on this team. Srinivas Shapuri is our tech lead, and Alicia Williamson is our curation lead. Funding support for this program, for the ImmPort program, is from the National Institutes of Allergy and Infectious Diseases. So thanks to all for their ongoing support of ImmPort. So for those of you on the call, some may be very familiar with ImmPort and others not so much. So we're going to briefly touch on exactly what is ImmPort. ImmPort, as you'll notice highlighted here up at the top right corner, ImmPort is celebrating their 20th anniversary this year, and ImmPort has had the opportunity to continue funding throughout these years, and we've really gotten to see how the data archive and data sharing through the research by the research community has evolved over the past 20 years. Exciting times ahead for data sharing, and we are really looking forward to supporting the immunology research community. So ImmPort is the immunology database and analysis portal. This portal is actually comprised of what we call the ImmPort ecosystem, which is 4 different portals. One is on the left-hand side is the data provider or private data portal. That portal really is set up for data providers and their private workspaces to submit data to ImmPort. We'll get into more details on what data submission to ImmPort is, but also how ImmPort can support fair data capture and sharing in the future. Shared data portal, off to your right, is the public-facing portal that anyone coming to ImmPort has access to. It facilitates the search and discovery of data that's available within ImmPort and does not require a login except for download. On the bottom right in purple is the ImmPort resources. That contains lots of different resources supported by the ImmPort program, including ImmPort gene lists, the cell ontology browser, and all of the ImmPort documentation supporting the ImmPort system. And that includes things like data submission documentation, also includes consortia web pages for the ImmPort team uses to feature different consortia that are supporting data or submitting data to ImmPort. We have online tutorials, and we'll be covering a little bit more of that later. ImmPort really focuses on capturing and transforming data to facilitate downstream data reuse. A lot of the studies in ImmPort are focused on, as one would expect, autoimmunity, infection, vaccine response, transplantation, and allergy. So where does the data from ImmPort come from? This slide shows you some of the programs that are continuing to submit data or have submitted data to ImmPort in the past. Obviously, lots of programs, data from a broad spectrum of data providers, Accelerating Medicines Partnership, the SERANET programs, loading their COVID studies, March of Dimes. The Civics program is actively loading their data at this current time in ImmPort. So across the 20 years of capturing data for sharing, this is just some of the programs. I'll be touching again on the Human Immunology Project Consortium. You'll hear me reference HIPSI in the future as well. Throughout this presentation, there are also key data providers for the ImmPort team. Off to your right, you'll see that annotation that ImmPort has received the Core Trust seal. We are now a Core Trust-worthy data repository, and I think we are on our second or third round of approval from the Core Trust seal team. You can find us in the r3data.org, as well as we're annotated as a recommended repository by Nature Scientific Data Listing and PLOS One. And we appreciate the listings for the journals, but that's not to say those are the only journals for which we're receiving data. Okay, we'll move on. So fair sharing. One of the things we definitely want to highlight is the ImmPort shared data browser. Through this browser, this makes it possible for anyone coming to ImmPort, regardless of whether or not they have registration, to search more than 900 studies that encompass a wide range of immunology areas, immunology research areas, species, and assay types. You can use the QR code on the right to go directly to the shared data browser. ImmPort, as this conversation will go along, we will show you how we focus and highlight fair data principles, and we ensure that your data is as fair as the ImmPort team can help make possible. ImmPort, again, has shared over 920 studies, which includes more than 100,000 subjects across 161 diseases, thousands of experiments, and literally millions of total results in ImmPort. So fair principles, really, what are the fair principles? Again, for the researchers or program staff from NIH, you've heard about fair principles quite a bit, and you probably know them quite intimately, so we're going to briefly touch on them here. Fair data principles are findable, accessible, interoperable, and reusable. And as we go through this presentation, we will touch on each aspect of the fair data principles and spotlight what ImmPort does to support each of these principles. ImmPort does strive to make data easily findable by everyone who does come to the ImmPort website. Easily accessible, the data is accessible through login, and it's easy to download in a multitude of formats. Interoperable, the data is transformed or annotated with numerous data standards, which then leads to interoperability or which then leads to reusability. The data submission process promotes fair data in many different ways, and this slide here is just a high-level 5-step view of the ImmPort data submission workflow. And we won't go into a deep dive on data submission today, but suffice it to say that through this 5-step workflow, your data that gets uploaded into ImmPort and then subsequently shared will be fair due to a variety of things that the ImmPort system does to support it. For one, on the bottom left, we have the study registration wizard, and that is a really nifty interface that users can begin to upload their basic study information into the ImmPort system. The user interface is very easy to use, very intuitive. There's a breadcrumb trail across the top that literally walks data providers through the data submission process for their initial study metadata. The ImmPort documentation, if you look to the right, we have lots of documentation when it comes to data submission and lots of documentation about the ImmPort data system overall. For data submission, we have workflows or decision trees that walk users through deciding which templates would best suit their research and the order in which those templates would be loaded. Each of the ImmPort templates has baked within it individual data standards through drop-down menus to further advance the fairness of ImmPort data. And at the bottom right is an image of the ImmPort data model, which is available through the ImmPort system, and it shows linkages between each of the different data elements within the ImmPort system. One of the things the ImmPort team and system does is make sure that each of the studies is appropriately identified, and that includes digital object identifiers, or as everyone knows them as DOIs. So upon data sharing, the ImmPort system will, ImmPort system and team will annotate each of the studies with a DOI as a persistent identifier, but within the ImmPort system, there are other persistent identifiers that we capture, and the example here are ORCID IDs. So for those data providers who are uploading their study personnel, ImmPort now accepts ORCID IDs. We also have PubMed identifiers to annotate each of the studies. For those of you who are submitting data and you have your publications, we will annotate each of the studies with the PubMed identifiers. It also makes it very easy to find data through searching with the PubMed identifiers. The ImmPort system also links out to other related information within the study details, and we'll get into that a little bit later. And that type of linking is like linking out to other resources to make the data more findable. So if users are within an ImmPort system and they're looking at studies that might have gene expression data in Geo, ImmPort will include those gene expression links out to Geo so that it's easy to find that data. Adherence to FAIR principles definitely increases the visibility of data within ImmPort. And one of the things we definitely want to point out is the cohort discovery tool that shows on your left. That is the ImmPort search feature in our shared data portal. Using the drop-down menus on the left, you can filter through all the studies within ImmPort and, adding specificity to each of those each of those filters helps you kind of fine-tune your search results. The ImmPort website also makes it very easy to export your search results in a table format. And then within the additional annotation that is available in each of the ImmPort studies, as I mentioned before, we link out to Geo. Vivly is another repository that is capturing ImmPort data. So if you happen to be at the Vivly resource and you're looking for studies, if you type in ImmPort, it'll return numerous studies from ImmPort for which that data is available in the ImmPort system. You can also find ImmPort data at the NIA data ecosystem, as well as through Google data search. And so ImmPort strives to not only make the data within ImmPort easily findable through the cohort discovery tool, we also work across the data sharing ecosystem, other teams, to make sure ImmPort data is findable through those other repositories or resources as well. I would like to highlight on the cohort discovery tool, there's lots of different ways to go ahead and search for ImmPort data. Research focus or condition or disease are the top two features that the research community uses to find specific data sets. So if you're looking for influenza data, you can go ahead and search under condition or disease. Or if you're looking for vaccine response data, then you would use the research focus. You can fine tune your searches through, selection of biosample types, assay types, and that type of thing. I highly encourage folks who haven't yet gone through and taken a look at the cohort discovery tool in ImmPort to please do so. It makes it very, very easy to discover research that aligns with your interests, and data download is a very simple process. So we just talked through how easily it is to find data within ImmPort. Data accessibility is also easily accomplished through ImmPort. ImmPort does have login requirements for folks coming to ImmPort who are choosing to download data. So each person who comes to ImmPort will be required to register for data. One of the other things is registration process is very easy. It's just your name and your email. And then we do ask a couple questions in the registration process so that we have a better understanding of how you learned about ImmPort. We also ask that when you are registering for ImmPort, you accept the data reuse agreement or the data use agreement, and that's publicly available when you go through the registration process. Registration and acceptance of the data use agreement is a requirement to upload or download data. So take careful view of that when you are signing up for ImmPort registration. Again, ImmPort registration is really, really simple, easy, and very fast. ImmPort application programming interfaces, our APIs are publicly available. You can find more details about the APIs and how to use those APIs within our ImmPort documentation. APIs make it very, very easy to query and access ImmPort data for download. For those of you who have those skill sets, please feel free to do that. The ImmPort user interface, the shared data interface that I was just showing before, the cohort discovery tool, there are easy ways to download data through that. There are little download buttons that you just click those buttons, and then that will redirect you to the shared data download portal. The shared data download portal, once you log in, will display the studies of interest that you have selected so that you can just click on those to download. The format, there's a couple different formats that are downloadable. Tab delimited is one, and there's a lot of familiarity with the research community with that format. So that makes data access easy. Interoperability, that's one of the key features of the ImmPort data and the data submission process, is to facilitate and promote data interoperability. So in the data submission process, as is shown here, this process, as I said before, is a simple 5-step process. It can get a bit more detailed as you start using the ImmPort templates. But off on the right, you'll see an example of an ImmPort template, the third image down. Those templates have baked within them ImmPort data standards. Those standards go a long way toward promoting data interoperability. The more standardized your data is, the more easily interoperable and potentially reusable it is. An example of the ImmPort templates is here. These are 3 different templates that we have: subjects, human subjects, immune exposure, and CyTOF. And back to the point of the controlled terminologies, the example on the right shows you what it looks like within the ImmPort templates, the drop-down menus that are available to help data providers standardize their data at submission, which overall increases the potential interoperability and reuse of that data. These types of drop-downs exist across the majority of our ImmPort templates, and it really does make it easier on the data provider to standardize their data right at upload. All of the documentation available in ImmPort is via this link. So we have a wonderful documentation set up in MKDocs. Easy to find all your data submission required data information there. The data submission process is outlined. The documentation shows how each of the templates, or shows a workflow for data submission. It also shows a decision tree, so you can make decisions on what you're submitting and in what order. One of the things the ImmPort system does is to leverage ontologies for controlled vocabularies in ImmPort. Again, referencing those ImmPort templates. So this is an example of just some of the ontologies the ImmPort system uses for linking, for annotating the different vocabulary that's used within the ImmPort system. It runs across immunology research and then beyond. We include the disease ontology for one, so that each of the diseases in ImmPort, listed in ImmPort, is annotated with a disease ontology ID. Obviously, protein ontology, cell ontology, and others. Further talking about interoperability within ImmPort, the next couple slides will be examples of data interoperability projects where research teams have used ImmPort publicly shared data and generated either additional new research, new discoveries, or in this case of 10,000 immunomes, a new resource. 10,000 immunomes was developed by the University of California, San Francisco team of ImmPort. The publication is there listed on your right. 10,000 immunomes consists of 85 studies retrieved from ImmPort shared data across over 10,000 subjects and 42,000 samples. The 10,000 immunomes project basically built a resource of controlled human study data, which could then be used as a foundation for future research. This is, the data is publicly accessible by the link on the bottom, as you can go ahead and see. You can also find 10,000 immunomes data available through a resource page in ImmPort. If you just click on the purple icon in ImmPort, click on the resources, then head to 10,000 immunomes. You'll be able to find some more information about that. And 10,000 immunomes really is a great example of what you can do with publicly available data. And in turn, this data becomes the foundation of other research from others because they can access and then download 10,000 immunomes data. Another example of interoperability that the ImmPort team has been working on is with other resources. The ImmPort program is pursuing the translation of ImmPort shared data into the HL7 FHIR standard. FHIR is the Fast Healthcare Interoperability Resource. The ImmPort team has developed a Happy FHIR server that's now publicly available, and that enables the research community to further query ImmPort in a different format. So if you're familiar with the FHIR server, or if you'd like to learn more about it, there is documentation on the ImmPort website about this FHIR program. The purpose of this not only makes the data available in an alternative format, which is FHIR, but it also takes the ImmPort shared data and broadens its accessibility to a wider community of researchers who are familiar with or comfortable working with data in the FHIR standard. One of the other examples of interoperability with resources across the research community is, in this example on the right, is a study the ImmPort team fondly calls Eureka. Eureka is a clinical trial study SDY 1644. And the screenshots there show you not only the basic study design, but it also shows you the ImmPort study links and how ImmPort will link out to other resources like clinicaltrials.gov, SRA, GEO, Bioproject, dbGaP. And this is one of the ways the ImmPort system can facilitate discovery of data sets related to the Eureka study, but that exist across other repositories. ImmPort subject and sample metadata, as noted before, when it exists in other repositories, and if it's listed in ImmPort, not only can you find it, but then you can go to those other resources, access that data there, and build a much larger data set than any one repository might be archiving. Reusability, the last part of FAIR data. ImmPort, one of the things ImmPort does to highlight where ImmPort data has been reused is in our publications page. Publications page is available under resources. You can find a listing of all import related publications. And in that listing of import related publications is the data reuse under type. So the import team constantly looks for publications that are coming out from the research community relating to import. We look at Google Scholar, we peruse PubMed, we use Publish or Perish software to find those publications that are referencing import. Oftentimes our data providers let us know when they've got a publication out, and we share data to time with that publication. But in other cases, the data is being reused and import team is not always informed. In those cases, we make sure we do environmental scanning so that we can find those publications, and we post them on the import publications website. We not only are data reuse papers listed there, but as you can see, we have over 1,300 publications at this point in time, and that was updated at the end of 2023. We have further updates that are coming, but of those 1,300 publications, in there, we also have publications that relate to primary analysis, data reuse, the use of import gene lists, which is a popular feature within import. So import publications further advances the findability of import data, but it also highlights or spotlights cases where import data has been reused. And a specific example of import data reuse is the systems vaccinology paper that came out in 2021 out of Bali Palandran's lab. And in this particular publication, they were looking at the impacts or the efficacy of the mRNA vaccine in humans. And they were comparing that, the COVID data to the data from shared data with an import across numerous studies in import. So if you look at the table on the bottom right, it lists a number of different import studies by their SDY numbers. And those vaccine response studies were collected together and used as a baseline for the vaccine response from Bali's lab in this publication. More information on this can be found obviously in the paper. And if you have any questions specifically about the data in this paper, please feel free to reach out to the import team. Another example of data reuse is a secondary use paper out from Sandra Andorf's lab out of Cincinnati Children's Hospital using the LEAP clinical trial study data, which involves peanut allergy infants at high risk for peanut allergies. And the secondary analysis of this paper specifically focused on this LEAP study data. The data was retrieved not only from import, but also from the immune trial ITN trial shared database and combined to go ahead and facilitate this research. And in this case, infants with a higher predicted predictability of peanut allergy was shown to benefit more from the early intervention of the peanut peanut testing. So thank you. In this case, Sandra Andorf and her team did reach out to the import team for assistance, and we helped guide them through data access and data reanalysis. Definitely, ImmuneSpace is another program who has actively used and continues to use import shared data. ImmuneSpace is out of the Human Immunology Project Consortium, and we much appreciate all their efforts in supporting the import program and reusing import shared data. We have active data providers from the HPSI program who continue to submit data to import. ImmuneSpace downloads HPSI data from import. They do so because the import data is formatted and it facilitates data reuse. So it makes it simple, easier for them to upload that and then use that data for their ImmuneSpace website. ImmuneSpace has a really nice web portal. So if those of you who haven't had a chance to take a look at it, please do. They have the ability to not only search for studies within ImmuneSpace, search for the HPSI studies, but they've also make it possible or have supported analysis on top of those additional studies. They have added data to HPSI studies that exist in import. So it's even a furtherance of the data that's in import. And then they also have a really nice data finder. In addition, studies in the immune signatures resource are archived through the shared data portal on ImmPort and also through immune space repositories. Over time, the immune space continually updates their repositories, as does ImmPort. So it's an ongoing connection between ImmPort and the immune space team. This just gives you so over the last three things I've talked about: the immune space, the LEAP paper reanalysis, and then out of Bali Palandran's the mRNA vaccine analysis. Each of those just provides you an example of all that can be done with using ImmPort shared data as a foundation for continuing an analysis, doing a reanalysis, or building a repository or a resource like the immune space program. One of the things we definitely wanted to touch on was AI and the use of AI tools across ImmPort data. The UCSF team took a look at reanalysis of ImmPort shared data, specifically in the 10,000 immunomes format, the CyTOF data that's found there, and ran that, questioned the data that's there through the GPT-4 tool, and ended up coming up with some interesting feedback from the GPT tool. And one of the things that did come up was age-associated differences in cell types were discoverable through the GPT-4 tool, and age and gender-associated effects on cytokines. This was something that the team put together rather quickly, and were able to facilitate not only reuse, show an example of reuse of ImmPort shared data, but also using another resource that was developed on the ImmPort shared data, like 10,000 immunomes. If you have, we do have, the ImmPort team does have a couple of people, Sammy Smith, who's not available for this call at this time, who are currently working on use of AI for supporting ImmPort in multiple different aspects, including taking a look at use of AI for facilitating data curation. If you have any questions about that, please feel free to reach out to the ImmPort team on what we're doing to use AI tools to advance the ImmPort system. A couple other things, I have to be aware of time here. A couple other things I do want to highlight, additional considerations for the ImmPort program is ImmPort data management and sharing plan resource that's available through the ImmPort system on the resources page. There's links out to other resources for further information, but we did want to go ahead and highlight that ImmPort does make has collected some information on the DMSP page, and it provides links out to other resources for those of you who are going to be completing grants and you're looking to develop your data management and sharing plan. Please take a look at the ImmPort resource. We have lots of guidance there that can help facilitate getting that done. And then thank you for considering ImmPort as a potential repository for your data management, for your future data needs. Again, I've mentioned numerous times ImmPort documentation. Please take a look at it for those of you who haven't had a chance to peruse the documentation yet. It covers all aspects of the ImmPort system, including new features. Data submission is obviously a highlight feature. Using the APIs, for those of you who are going to be searching for ImmPort data and are comfortable using APIs, we have documentation to support that. Online tutorials, as well as a newsletter sign up. ImmPort does have a newsletter, and if you'd like to be interested, please take a look at that and plan to sign up for the ImmPort newsletters. They go out roughly once a month. And then also in our resources is we do have a collection of past webinars and a listing of upcoming events. And that's it. I'd like to open up for conversation or Q&A if anyone has any questions. No questions? Okay, I would like to note that the ImmPort team just had a data release. Oh, I see one thing in Q&A. ImmPort did just have a data release. We are now doing monthly data releases. So for those of you who are data providers or potential data providers, we are conducting monthly data releases. And Zelenel, can you pull up the Q&A?
I can.
Thank you.
So there is a question, is there a way to share studies between workspaces?
Good question. Do we have, so I can't actually see who's on the call right now. Do we have Matt on this call or Ziping?
Yes, so the question, share studies across the workspace. So it's actually you can, so what kind of the share is actually you want to have? If you say, okay, I want to have share subjects, right? And then probably you cannot, you need to create, because within each workspace, the subject can be shared. So you submitted one study, now you say, okay, I want to have another study, and use the same subject. You can share within the workspace. But currently you cannot share the subject from the other workspace. You need to create the same subject within this workspace. Yeah, currently this is the, I think it's not shareable. Yeah. But you
And I would say, please feel free, if you haven't already reached out to the help desk with this specific question, and we will try to get back to you with specifics on what we might be able to do to facilitate sharing across workspaces. But currently, per Ziping's feedback, that's not currently possible.
Yeah, so sharing has the different levels. Some people just say, okay, I want to see all the studies is actually the similar studies across different workspace. This is certainly you can. Yeah, you can search based on our facet search page. You click some kind of the domain or some kind of the same analytic assays. You can get all the studies, regardless of in which space. You can get all of them. Yeah.
Okay, thank you, Ziping. But again, for the person who asked the question, please reach out to the help desk, and we'll try to get you some specific details on what we can do to support.
Thank you, Sabine. We do have two more. There's one that says, can we access tools for meta-analysis at ImmPort?
So ImmPort does not develop tools specifically. We do have a suite of tools, but they're not geared for meta-analysis. We have a suite of analysis tools that are specific for flow cytometry, and those are in ImmPort Galaxy. But for a meta-analysis, we have tutorials on ImmPort that you can go ahead and peruse the tutorials on ImmPort. I believe we have one tutorial focused on meta-analysis, but please feel free to take a look. But no, we don't have specific tools. Any other questions?
Yes, there's another one that says, is the GPT integrated in ImmPort, or do we have to export it into one of the applications? And then there's a follow-up question to that that says, can applications like ChatGPT be used to fill out the templates?
Good questions. No, ImmPort does not currently have GPT within the system. This is something the team has done on the side. But going forward, we're looking at lots of different options for the use of AI and AI tools to facilitate, lots of things across ImmPort, including data curation. So the second question was about using AI tools to facilitate upload or completing templates. Is that correct?
Yes, completing the filling out the templates.
Yes, well, we've had that conversation before, and it is something the ImmPort team is going to be looking at. There is potential opportunity using AI tools to do just that. And if we could streamline data submission, the ImmPort team would be very interested in doing that. So it's something the ImmPort team will be looking at in the future. Okay. If we don't have any other questions, I did want to make a note. The import team will be at AAI coming up in this May. There's 2 presentations from the import team, 2 posters, presentations from the import team. So for those of you who might be attending AAI, we will have 2 posters. Also, Dr. Toole Butte will be one of the panelists at the NIAID supported symposium at AAI. We also have a focus workshop coming up. So focus is being hosted in June this year. I believe it's in San Francisco, and the import team has been invited back yet again to host their focus workshops. These workshops are focused on big data. And I believe this year we are going to be looking at using AI tools for potential data reanalysis. So if you're going to be at Focus, you definitely want to go ahead and plan to register for the ImmPort workshop on AI. All right. I thank everybody for participating in this webinar. As I mentioned at the outset, the ImmPort team is beginning a webinar series. And so if you have comments on what you would like to hear about in future webinar series, we would greatly appreciate either your input on the Q&A now, or in the future, if you'd like to reach out to the help desk and there's something specific you'd like the ImmPort team to cover in a webinar, please let us know. Again, we're just starting this for the ImmPort team, so we're very interested in addressing questions and comments the user community is most interested in. So I'd like to thank everybody for the time you spent with us today, and we look forward to seeing you again at future ImmPort webinars. Thank you very much.